Okay, so let's talk about the phylum Annelida. So let's check out. Let me find my pen. Sorry about that. Okay, so here we are with our annelids. So reminder, we're still in this Lophotrochozoan clade. So the Annelida are the segmented worms. Whoops, getting ahead of myself. So these are found in freshwater, marine, and terrestrial damp environments. They are coelomates, so they have that true body cavity. They have highly segmented bodies, and this division is also seen in the coelom. The segments are largely controlled independently by nerve centers in each segment. They exchange gases across their skin, so it's important they are in a moist environment because in order to exchange gases, it has to be a wet membrane, so the skin must be wet. So we are going to talk about two classes, the class Polychaeta and the class Clitellea. So let's dig first into the class Polychaeta. So these are almost all marine and highly mobile, although there are some that are sedentary. We find them swimming among the plankton, burrowing on the sea floor. Some are immobile tube dwellers, as you see here. Some are highly mobile organisms that can be predators, grazers, or browsers. Many have a pair of paddle or ridge-like appendages called parapodia. These parapodia, or singular parapodium, each parapodium has chaetae, which are bristle-like extensions or projections made of chitin. So it's a little hard to see, but you'll see this much better in lab. So here, this little paddle-like appendage is a parapodium, and then we've got these bristle-like chaetae coming off them. So for sensory structures, we typically have one or more pairs of eyes and tentacles on these organisms, although this is going to vary from group to group. We are going to look at a number of different organisms that are in the class Polychaeta when we're in lab. So hopefully that'll help with some of the diversity we see here. Class Clitellata. We're going to actually look at two distinct groups, two distinct clades within this class. Aligiochaetes and the leeches. So most members in this class have far fewer bristles on each segment than we see in the polychaetes. So poly means many, so they've got, the polychaetes have lots and lots of those bristles, whereas here we don't, within this class we don't see as many. They are typically slower, less mobile, so we have a lot of slow burrowers or tube dwellers, and we're going to talk about the two major clades. So in the Aligiochaetes, kind of our poster child that you're probably all familiar with is the earthworm. So here we are lacking parapodia, and we do have some chaetae on the segments, but far fewer than we see in the polychaetes. And we have no eyes or tentacles. So for an excretory system, we have something called metanephridia in each segment that will filter fluid and excrete it externally. They eat soil and will extract nutrients from their soil. So let's take a look, because this is something we're going to look at in lab. We're going to look at the earthworm's anatomy quite a bit in lab. And so you'll see this picture again for sure. But here is an example of the metanephridium that we're going to be finding in each segment that will help in filtering fluid and excreting waste externally from the organism. Again, we have a concentration of nervous tissue at the anterior end, and we have these ventral nerve cords going down the length of the organism. You can see that we've got a highly segmented body, and that these segments are actually through the entire cavity. So we have the septum here is dividing these compartments. So we have a complete digestive system with a mouth at one end and an anus at the other end. And we also have, of course, reproductive structures. And earthworms are actually hermaphrodites that will cross-fertilize. Two individuals will align in opposite directions, which is what you're looking at here. They will exchange sperm, and then they will separate. And this structure, called the clitellum, is a specialized reproductive structure that can be where we can put our fertilized gametes and shed them out into the environment. And we'll talk a little more in lab about this whole process. Also, some members can reproduce asexually via fragmentation and regeneration. I know when I was a kid, that's something people used to do. It's really horrible to think about now, but we would cut an earthworm in half and see that it would regenerate. I don't recommend it. I don't think that's a kind thing to do, but it is a method of asexual reproduction. All right, here's a horrifying image. So here are some leeches. So something about the leeches in this group, although we see the evidence of segmentation on the outside, the segments are not internally divided like we see in the earthworm. So these are primarily ectoparasites that feed on the blood of other organisms, and so they have an anterior sucker that usually contains some kind of mouth parts that will allow the organism to cut into its host's flesh, and so it can suck the blood. And then, not shown here, oh, actually, you can kind of see it here, we have a posterior sucker that would just be for hanging on. So the anterior sucker would be for sucking up the blood. This is just kind of a hanging on sucker usually. So they use this blade-like jaw to slit the skin, and they secrete enzymes that will eat a hole in the skin is another way that they do it. Some organisms do it that way. The leech has an anesthetic in its saliva and also secretes another chemical that prevents blood coagulation, so the blood will keep flowing. And these leeches can actually take in so much blood that they become, several-fold times their original size before they just can't take anymore and they drop off. Historically, they've been used medicinally for, bloodletting and things like that. Now we realize that bloodletting for the sake of bloodletting is not a good medicinal practice, but they actually are still used in modern medicine in some contexts to, I have heard of them being used in the cases of if you have surgery and you need to reduce inflammation around a site that perhaps has been stitched up, sometimes that can be useful in getting things to heal, to kind of get rid of some of the excess inflammation and things, which it looks like maybe they're trying to reduce some of the inflammation and things in this area. I'm not sure exactly, but I would prefer not to do it to myself. I've also heard that there are some modern beauty treatments that involve the application of leeches. I'm not for it, but I guess some people are. So, you know, make your own decision there, but it is a parasite, so I can't recommend it. Anyway, moving on to a highly impressive phyla by any means, by any measure, and that is the phylum Mollusca. Okay, so this is the most diverse group of Lophotrochozoans. These include a lot of animals you've probably seen or heard of, such as snails, slugs, oysters, clams, octopuses, squids. They are all soft-bodied. Some secrete a hard protective shell, usually made of calcium carbonate. There are some members, including the slugs, squids, and octopuses, that have a highly reduced shell that is internal, or, in the case of octopuses, have lost the shell altogether. Their digestive system is an alimentary canal. Their circulatory system is varied. So it is open in most, but one group called the cephalopods, which include things like squids, have a closed circulatory system. So what do I mean by open versus closed circulation? I guess I should explain that, huh? So in open circulation, we don't really have what we call blood, because the term blood is contained within a vessel. So we talk about a fluid called hemolymph that will, and this is an example in an insect, there are some sort of truncated vessels and that will allow the pumping of this hemolymph, but it enters kind of this open area where it can just sort of percolate between cells and tissues, and then flow back into some abbreviated tubules where it can be pumped by a heart again. So it's open because it's just, well, it's open. There's this area where it can just sort of allow every, tissue in here to just be bathed in the circulatory fluid, which in an open system we call hemolymph. Now, closed circulatory system, we differentiate, and we don't say we just have hemolymph, we say we have blood, and the other kind of fluid, such as the fluid you might find in the body cavity or surrounding the cells, is interstitial fluid. And the blood stays within some kind of vessel. And there can be exchange of things like nutrients and waste between the blood and the interstitial fluid, but the blood doesn't actually leak out into the cavity where the interstitial fluid is. So that is the difference between a closed and an open circulatory system. Another thing about mollusca, I wanted to mention the nervous system. Generally, the nervous system is simple, very simple in most, but pretty complex in the cephalopods. So the cephalopods are kind of special in the mollusca phylum. So this is the generalized molluscan body. there's a huge amount of variations on this body. So you might look at this and think, this looks nothing like a squid. Okay, I agree. It really doesn't look a lot like a squid. But, believe it or not, a squid or an octopus or a slug or a clam all actually have variations of this basic body plan, and in particular, 3 common structures. A muscular foot that is, did I write foot? That's funny. A muscular foot, so this is typically a very muscular structure, often used for movement. We have the visceral mass. I'm going to actually number these. These are the 3 structures. The visceral mass that contains most of the internal organs. And we have a mantle. So a mantle is a fold of tissue that drapes over the visceral mass and in most organisms will secrete a calcium carbonate shell. The mantle may form a fluid-filled chamber called the mantle cavity that typically will house the gills, the anus, and excretory pores. So let's take a look at these structures. Okay. So this whole area structure down here is the foot. It's just this very muscular region, usually on the bottom, and in this simple molluscan body, it would be used to help the organism move. In different groups of mollusks, this feature has been modified in different ways. The visceral mass is the vast majority of what you're looking at here. So this would be our this would be our visceral mass, containing most of the internal organs. The mantle is this, in blue, this fold of tissue. The mantle is what's going to secrete the shell. Now, as I said, there are some mollusks that have a highly reduced shell or no shell at all, but generally speaking, this is what happens with the mantle. So sometimes the mantle will overhang the visceral mass and create this little pocket that fills with fluid called the mantle cavity. And oftentimes it is here that we find the gills, the anus, or other kind of excretory pores. Many, not all, but many have this little organ called a radula that can rasp and scrape up food particles. So they don't all do this. Some types of mollusks don't have this, and rather they feed by filtering through their gills. So as I said, this is a really diverse phylum, so we're going to see a lot of variation on these structures. But you'll see some form of these structures in all of our mollusks. So let's talk about the chitons. These are probably the closest to this sort of classic mollusk structure, kind of. I mean, not perfectly, but it's the right shape. So to me, it always seems the closest. So, class Polyplacophora. We're going to be talking about several different classes within the phylum Mollusca. Commonly known as the chitons. right. Here's our little Mr. Keaton. So some features we've got here is we have these 8 dorsal plates along its back. And you can't really see it very well here, but there is a extension of the mantle, and we'll see it better in lab. So if I was looking at this thing sort of from the belly, I'd see the organism, then there'd be this kind of extension of the mantle that we call the girdle. The foot contains a suction cup that helps it cling to the rocks, and it has a really nice pronounced radula that allows it to scrape algae from the rocks. So usually this guy is found kind of clinging to rocks where it scrapes up algae and things like that. And we'll look at some of these guys in lab. All right, let's look at the gastropods, snails and slugs. Class Gastropoda. Big group, 85,000 species, most marine, some freshwater, many are terrestrial. They have a really interesting developmental process known as torsion. So in torsion, so this is a developmental process. In which the visceral mass rotates 180 degrees. Causing the anus and the mantle cavity to rest above the head. So now this might seem really weird. It does seem really weird, but it's actually kind of a really good idea. Because what this means is that you can have a shell with just one opening rather than 2 openings. So it reduces the likelihood that something's going to get into your shell other than you. So it actually makes good sense that this would be something that would happen. So here's an example. So here's the mouth. And then again, this process of torsion caused it such that the anus and mantle cavity are up here by the head rather than being back here like you might expect. So many do have this radula that they can use for grazing plants, but some are predatory and actually will bore holes into organisms, for example, maybe through the shell of a clam or other bivalve with their radula. Some, notably the cone snails that we'll look at in lab, not live ones, have venom in the teeth of their radula that they can actually eject into their prey to subdue them. They're quite poisonous. All right, so let's talk about our next class, Bivalvia, the bivalves. So, here's my little cartoon of a scallop. So these are all aquatic. They get called bivalves because they have 2 halves of their shells that are hinged together and shut by muscles. No head, no radula. Some have eyes and other sensory structures like tentacles along the edge of their mantle. They are typically suspension feeders that will circulate water over their mantle cavity using siphons. So usually we have what's called an in-current siphon and an ex-current siphon. So they can take in water, circulate it over the gills in their mantle cavity, and do suspension feeding that way, and then send it back out. Most are sedentary, but some can actually move either with a muscular foot or by flapping of shells, which is what you're seeing here in this little animation. So this is a scallop that I think this is sped up a little bit, probably. So this is just repeating over and over again. Otherwise, this guy would be covering quite a bit of ground. But they can flap their shells and relocate themselves in some cases. So it's kind of neat. So we'll also be looking quite a bit at some bivalves in lab. So just kind of make note of a few structures here. So we're looking at sort of on the half shell here. So we have our mantle here in blue that would have secreted that shell. So this mantle would cover the entire inside of the shell. You're not really seeing it here. There's our mantle of the lower shell, not seen. We've got our heart, we've got an intestine, including an anus here and a mouth on the other side. Here's a digestive gland. Here are the siphons that will allow water to come in so we can filter feed using the gills. Here is a muscular foot that could possibly be used, to open and close the shell. Depending on its ability to extend out of the shell, could also maybe be used for movement. Okay, class Cephalota, the cephalopods. Class Cephalopoda. Octopuses, squids, and nautiluses. So this is a really cool group where we're going to see some really extreme modifications of the basic molluscan body plan. So these guys are all active marine predators that use tentacles to grasp prey, and they typically bite with a beak-like jaw and will immobilize their prey with poisonous saliva. The foot, the classic molluscan foot, which pretty much was just a lump of muscle, has been modified into a muscular excurrent siphon into arms, tentacles, both of which can have suckers. The arms tend to have suckers all over them, whereas the tentacles would typically just have suckers at the ends. So let's talk about this muscular excurrent siphon. So this muscular excurrent siphon, now I said that these were active predators, so they're not using this to filter feed. What they use this for is to control the amount of water volume in their body. And so basically what they use it for is jet propulsion. They can fill their body with water and then rapidly push a jet of water out through this siphon, which can give them jet propulsion. So it's pretty neat. So the shell is typically reduced and internal, or absent. The notable exception would be the nautiluses that have a external and chambered shell. These guys have a closed circulatory system. So this is sort of unique. The other phylum Mollusca animals had open circulatory systems. And this closed circulatory system increases efficiency of transport, allowing for rapid movement in response to the environment, which makes sense because these guys are active predators. They need to really be able to respond to their environment. They have well-developed sense organs and a complex brain. Their learning and behavior is really quite advanced. Fossil cephalopods called ammonites were the dominant marine invertebrate predators for hundreds of millions of years. And they got absolutely huge, up to 2 meters in diameter, which was really big back then. They went extinct in the Cretaceous with the dinosaurs, but I don't have a picture. Let me go find a picture. Okay, so there's a picture of one of these. So yeah, big. And probably the closest thing we have comparatively is the chambered nautiluses, although they're not exactly the same. So yeah, pretty neat. Okay, so what else did I want to say about these guys? I know what I wanted to say, so let me get rid of these guys for a second. What I wanted to say is that our squids, whoopsie daisy, hot mess over here. Okay, our squids tend to have the reduced internal shell. Our chambered nautiluses all obviously have the external chambered shell. And our octopuses, no shell. Okay, so that's kind of how it breaks down with the shells in the phylum Mollusca. All right, believe it or not, we made it through our Lophotrochozoans. I know. So we didn't talk about every single one of them, but we did talk about the oops, I got my wrong end of my pen. The mollusks, the annelids, the flatworms, the rotifers. So we will also be looking at all of these animals in the lab. In our next video, we're going to pick up with the ecdysozoans, which is another absolutely tremendously huge clade. So we're going to take a little break because it's a big clade, and see you soon.